Charles Darwin. The scientist who contributed most to our current understanding of evolution was Charles Darwin. Upon graduation from university, Charles Darwin took a position as a naturalist on a ship called the H.M.S. Beagle. The main purpose of the Beagle was to map out the coastline of South America. Darwin was on board the Beagle from 1831 to 1836 to record all natural and geological characteristics of the landscape he saw. He was only 22 years old when he embarked on the journey, so he spent half of his 20s on the Beagle sailing around the world. Journey of the H.M.S. Beagle from 1831 to 1836. Started here in Europe, in England, and then Charles Darwin sailed down to map out the coast of South America, stopped here at the Galapagos Islands, and then finished his journey across the Pacific Ocean, the Indian Ocean, back up to England. Darwin arrives in South America. Roughly 1/3 of the way into the journey, Darwin spent most of his time inland collecting thousands of fossils, specimens, and living plants and animals from South America. He sent all of his collection, thousands of specimens, back to England by ship while still on a journey. And since he was so observant, he took all these journal entries of his collection. He even wrote a book on barnacles. A very big book on barnacles, by the way. The Galapagos Islands. In the 4th year of the voyage, the H.M.S. Beagle reached the Galapagos Islands off the coast of Ecuador. Darwin found many plant and animal species that were similar to the species on the mainland of South America. He compared the island and mainland species and found that the island varieties had different adaptations from the same mainland species. So all the species he observed on these Galapagos Islands were slightly different than the ones found in Ecuador. One example, marine and terrestrial iguanas. While in the Galapagos, Charles became fascinated with marine iguanas. He noted the differences in adaptations between both species. The terrestrial iguana on the left was found in mainland South America, and the marine iguana in the Galapagos is pictured on the right. Marine and terrestrial iguanas. At first glance, these two lizards may look very similar, but Charles Darwin was very observant, and he noted the differences between these iguanas. The terrestrial iguana in mainland South America had a round tail, lived on land, had a fear of man, did not swim, and had smaller skin flaps on its back. Marine iguanas in the Galapagos, however, had a flat tail, dove to the bottom of the ocean to eat algae on the rocks, no fear of man, has large skin flaps on its back, and longer and sharper claws. Physical adaptations of the marine iguana. Flat tails allow it to swim to get its food. Large skin tags on its back provide more surface area to gain heat from the sun more quickly. Blood vessels constrict when it is in the water to minimize heat loss. Longer and sharper claws allow it to cling to the lava rocks when in the water so they don't get carried out to sea. And longer and sharper teeth allow it to scrape the algae off of rocks. Darwin's finches. While in the Galapagos, Darwin also studied small songbirds called finches. He studied their feeding habits and quickly realized there was a connection between the type of food they ate to the beak size and shape. So beaks, very important to birds because they don't have hands like we do, so they need the beak to obtain food and also handle it. He saw there were many varieties among the finches. Some had large beaks, small beaks, thin beaks, and thick beaks. Finch beak size and function. Each beak is adapted to eat a certain type of food. So the bird with the thin beak here, that beak is for probing. It feeds on insects found in trees and the tree bark, sort of gets in there between the crevices of the bark. And we have another bird that has a probing bill for grabbing twigs and using that twig to get insects from a cactus. Or the grasping bill, an insect eater also found in trees. And then the crushing bill. This bill is shorter and thicker, so it has more leverage. And since it's thicker, it has more durability crushing cactus seeds, which are very hard on the outside. Click for a Darwin finch video. Harriet the Galapagos tortoise. One of the living animals Darwin brought back to England was a young tortoise he named Harriet. She was only 5 years old. Harriet lived with Darwin as a pet, then lived in an Australian zoo owned by Steve Irwin, the crocodile hunter. Harriet died at the age of 176 years in 2006. Darwin's return to England. Upon Darwin's return to England, his collections were hailed by the scientific community. Most scientists, let alone the general public, had ever seen or heard of the organisms he brought back. He immediately began to send out specimens to other scientists for examination. He needed help because of the sheer volume of what he had collected. He started to piece together the evidence to determine the mechanism by which evolution happens. This work took years. Darwin's evidence for evolution. Darwin used 4 categories of evidence to develop his theory. 1. Comparative anatomy, including homologous structures and vestigial organs. 2. Comparative embryology. 3. Fossils. And 4. Biogeography. Comparative anatomy. Comparative anatomy is the comparison of different body structures in different species. Anatomical similarities give signs of common descent or that species are related closely through evolution. So for example, we have the Asian elephant on the left, the African elephant on the right, and we can see a clear difference in the size of their trunks, and also the Asian elephant has more of a pronounced skull. And then the more similarities there are, the more closely related the two species must be in evolution. So these two elephants more closely related to each other than say the elephant is related to a giraffe. Homologous structures. Darwin noticed that animals have similar body plans and structures. He saw that the main differences amongst animals were in the forearm and hind limb areas. However, forelimbs of different animals are made up of the same exact bones, only the bones are modified to fit the environment of the organism. These are known as homologous structures. So here we have a diagram of different forelimbs of various organisms, including humans. And so if you look at these bones down here, these are the phalanges. So for humans, the phalanges are our hands are used for grasping things. And for lizards and cats, the phalanges are used to walk on. In a whale, the phalanges are used as a flipper for swimming. And then in a bat, the phalanges are modified to form the structure of a wing for flying. So all these bones are the exact same bones, part of the similar body structures, but they are modified for the specific purposes of these organisms and the environments that they are in. Vestigial structures. Vestigial structures are structures on an organism which have lost function and atrophied or shrunk through time. The body part had a function in an ancestor, but through modifications and evolution, the body part is no longer useful. So for example here, we had the flightless cormorant, which is the descendant of a bird that can fly. But over time, it adapted for different purposes, didn't need to fly anymore, and so the wings have just shrunk down and atrophied. Vestigial bones. Whales have a femur, or a thigh bone for a leg, and a pelvis, or a hip bone. Both of these bones are associated with walking. But we know that whales don't walk, they swim. So it is believed that the whale evolved from an ancestor who was able to walk on land. And then as this ancestor became more associated with the water, it was more advantageous for this ancestor to swim and to develop other structures for swimming. And then consequently, the bones associated with walking just became smaller and smaller. And then here they are today. Similar story with the snake. Snakes have hips and leg bones as well, but they've evolved more for slithering on the ground, and then these bones are no longer useful and they just shrink over time. A vestigial organ, the human appendix. What is now the human appendix was once an extra pouch to help digest food back when our ancestors were herbivores and ate mainly plant material. Since then, man has developed into more of an omnivore, eating both meat and plants. So the pouch has lost both its function and its original structure, We can see that when we compare our appendix with the appendix of other vegetarian herbivore animals, the opossum, the kangaroo, koala, rabbits, and zebras. Highlighted in purple, we can compare the size of the appendixes relative to the size of the whole GI tract, and we could see in the humans it's just very small compared to the zebra, for instance, has a much bigger appendix because zebras are just herbivores, and so the appendix has more enzymes and other chemicals that help break down the grassy materials that they eat.